What we are going to be talking about today is soy glycolins and how they regulate transcript abundance in the mouse brain. Glycolins are produced by soy plants in response to stress. They have anti-estrogenic activity in breast and ovarian cancer cell lines in vitro and vivo. Several recent studies have reported that mixtures of the isomer glycolins 1, 2, and 3 affect mammalian physiology in vivo and vitro, and what emerged from that is a complicated and incomplete picture of how GLY affects mammalian physiology. The findings also indicated that GLY opposes the activity in estrogen-responsive tissue, such as the uterus. GLY suppressed E2-mediated proliferation of MCF7 human breast tumor cell lines in vitro, and GLY inhibited E2-stimulated growth of MCF7 breast cancer and BG1 ovarian cancer cells, implemented as xenograft tumors in overactinomicized nude mice in vivo. However, other studies have concluded that GLY can also exhibit weak estrogenic activity, which appears to be enabled GLY to promote the proliferation of MCF7 breast tumor cells in vitro. Glycolins exhibits mechanisms of action not involving estrogen receptors, which are also known as ER signaling. To date, effects of GLY on gene expression in the brain are unknown, and this was the first experiment performed on the brain using GLY. There also have been other studies that have involved kidneys, but this was the first one that involved the brain. The next figure I'm going to show you points out the effects of GLY known in vitro and vivo that are ER-mediated versus non-ER-mediated. It also is pointing out the results of several previous studies showing effects of glycolins that depend on blocking the estrogen receptors, also known as the ER-dependent, and effects that have nothing to do with the estrogen receptors action, also known as ER-independent. For example, breast cancer tumors that have estrogen receptors, also known as ER, shrinks when exposed to glycolins. Because estrogen receptors make those tumors grow and GLY blocks ER functions. However, the triple-negative breast cancer cells do not have estrogen receptors. So when glycolin shrinks those tumors, it can't, through its action at the ER, since those cells don't have ER. The female mice, once they had their ovaries removed, the scientists had to wait a period of time for the mice to recover from the procedure because it's an intense procedure. The mice were then implanted with estrogen, and the placebo were slow-release pellets. The following, there were 11 days of exposure to GLY or vehicle IP injections. On the morning of day 12, mice were then weighed and then euthanized by decapitation under complete isoflurane anesthesia, and trunk blood was collected. Brain hemisphere restored in RNA overnight. Because the mouse gene 1 ST array covers 35,556 transcript clusters, there is a considerable multiple testing burden associated with analyzing gene-level data from this platform. In order to minimize this burden and avoid conducting tests on genes that are unlikely to be expressed in the brain, the scientists then removed transcript clusters whose mean expressed across all chips was less than and equal to the mean of the first quartiles across all chips. In addition, the scientists removed transcript clusters that were not part of the main design of the array. The scientists removed all control and unmapped transcript clusters prior to conducting statistical analyses. Upon applying these filters, 22,056 transcript clusters were available for the significance of testing. The scientists examined all arrays at the probe level by inspecting box plots, histograms, and more. They used gene expression estimates in standard arrays obtained from the PLM to render relative log expression and normalized unscaled standard error plots. The scientists then performed a microarray on total RNA extracted from whole brain hemispheres, and then identified differently expressed genes by a 2x2 factorial ANOVA with a false discovery rate of 0.2. This next graph, it's a heat map, shows that estrogen groups often respond opposite to the glycylation-only group, and when both are used together, the pattern on the chart looks more like an E2 group than the GLY group. That means that the estrogen is the stronger signal. Each column represents the female mice. Each row represents a gene. Gene expression is measured by the amount of mRNA for the gene in the brain. The average mRNA expressed for the gene across all groups is in black. Red indicates increased expression and also meaning that there are more mRNA copies are produced for the gene. Green means decreased expression and also means fewer than average amount of mRNA copies. In total, the scientists identified 33 DEGs with a significant E2 effect, 5 DEGs with a significant GLY main effect, 74 DEGs with a significant GLY and E2 main effects, and then 162 DEGs with a significant interaction term. When going over all DEGs, it revealed that transcript abundances were similar between the estrogen and GLY and only estrogen treatments. However, after GLY-only treatments, the gene expressed was distinct from both of these treatments and was characterized by higher transcript abundance. The results suggest that whether GLY acts in the brain through ER-dependent or ER-dependent mechanisms depends on the target gene. E2-exposed animals had nearly 47 times the circulated E2 levels in non-E2 exposed animals, whose plasma E2 levels were at or near the lower detection limit of the assay. Collectively, the results show that animals in the E2 groups had significantly higher plasma E2 as compared to non-E2 exposed animals, and that GLY may lower circulating E2 concentrations in animals exposed to estrogen.